beginning with Dr. David Muddiman. Dave is a professor at North Carolina State University in the Department of Chemistry. His research encompasses the development and application of novel mass spec platform technologies, systems biology, and environmental science toward better understanding human disease. In addition to the hundreds of talks delivered worldwide, his group has published over 300 manuscripts and has received 6 U.S. patents. Among his accolades, Dave has bestowed the 2023 Donald Hunt Distinguished Contribution in Proteomics Award at US HUPO. We are quite pleased to have Dr. Muddiman provide a glimpse into some of his research endeavors in a talk entitled The Critical Role of Stable Isotopes to Unravel Complex Biological Questions.
Okay.
Dave, if you'd like to take over, the floor is yours.
Okay, thank you, Andrew. It's a pleasure to be here this morning and to tell you about 3 short stories about how we're using stable isotopes, but not in the things we've done in the past, but doing things moving forward with a platform in mass spectrometry imaging. So the technology that we use is something developed in our laboratory called infrared matrix-assisted laser desorption electrospray ionization. And so in general, it's an electrospray source where the charged droplets come across this interface and enter into the mass spectrometer, and we have a tissue sample. And here you notice it's actually a piece of skin, and so we can do 2-dimensional imaging and 3-dimensional imaging with this technique. What we do is we simply have these charged droplets above the surface. We can use endogenous water, or we can add an exogenous ice matrix on the top of the tissue by condensing water out of air. We fire the laser, neutrals partition into these charged droplets, and just like in electrospray ionization, we basically get a wide diversity of analytes into the gas phase. This basically combines the advantages of electrospray and MALDI into one technique, and so we can use this now for mass spectrometry imaging. And for those people that are interested, there's a recent review article in 2021 in Mass Spec Reviews covering up the work up until that time. With this technique, we've done intact proteins. You know, this is native mass spectrometry, so we're seeing intact proteins in the native state. We do glycan imaging, lipid imaging, neurotransmitters, and metabolite imaging. So today I'm just going to tell you a few short stories about how we use this technique. One thing is, if you're not familiar with mass spectrometry imaging, we have a collaborative tutorial series that was published in the journal Mass Spectrometry Imaging on different aspects of mass spectrometry imaging, and a 5th one is coming out soon on the relative and absolute quantification of biological molecules in the field of mass spec imaging. So the first story has to do with glutathione and its elevation in tumors. And so the GSH is an important antioxidant and regulator of redox homeostasis. It promotes tumor progression and chemotherapy resistance, and we're trying to understand this. And so over here, what you see is a hen ovarian cancer sample. So on the left, this is healthy ovarian tissue. On the right, you can see it's lit up pretty bright with upregulation of glutathione. And so the glutathione pathway, you could make it in many different ways, several different ways, I should say. And then the reduced form is present in 98% of cells. It's composed of 3 amino acids, glutamate, cysteine, and glycine. And so what we're going to do is we're going to use the biosynthetic pathway, and we're going to incorporate in stable isotope glycine into the formation of glutathione in real time in a mouse model. So taking a step back, this has to do with functional mass spectrometry imaging, so we can get towards spatial fluxomics. So in LCMS and NMR, you can do molecular fluxomics. You get multiple time point sensitive information, but no spatial information about where is the glutathione present. You can also use functional MRI and PET scanners, but it has limited spatial resolution and molecular sensitivity. So in molecular imaging, we can do mass spec imaging, IHC, and so forth, but we end up with a single time point. So we get the molecular information, but we don't get the we only have a single time point, and that's not going to tell you whether glutathione is being upregulated, downregulated, and how it's changing as a function of the tissue type within the very heterogeneous samples that we look at. So we wrote a review article on this on functional mass spectrometry imaging and JPR, so you can look that up if you're interested in learning more about functional mass spec imaging. The idea here was, could we leverage mass spec imaging to measure function? So we want to measure multiple time points, but we didn't want to have to sacrifice multiple animals. So starting all animals out at the same time, and then basically infusing them with a single isotope and going forward. The problem with that is that you have a lot of biological variability associated with those type of approaches. So we came up with this idea called MIST, or multiple infusion start time. We bought 3 isotope logs of glycine from CIL, and these are shown here. And then what we can do is infuse these at different times. So T1 is right at the beginning, and then we can do one at T2 and T3, then we can harvest the tissue. And then we can actually resolve these different stable isotope labeled forms of glutathione because they have distinctly different M/Z values, which we can resolve with a HRAM, with a modern mass spectrometer. And so we have, like I said, a distinct labeling pattern, and we get multiple time points, but from a single tissue. So we remove that biological heterogeneity from the process. So this is in collaboration with Michael Gamsick and a former graduate student of mine, Allison Mellinger, who did this work. We take the MIST with the stable isotope labeled glycine. We do tail vein injections. We then put the tissues on a coated slide. So this is homoglutathione. This is only found in plants, and so we can use that as a normalization standard to get better reproducibility across the tissue images. Then we do imaging, and then we do data processing and visualization in a program called MSI Reader. And so this work here made the cover of JPR. It's a really nice article. It was laying the foundation for this idea, so it's really a proof of concept paper, and we did this with liver tissue. And so this is showing you the different 3 forms of the stable isotope labeled 1, 2, and 3. This is of the glycine, but ultimately, of course, it makes it into the glutathione. So these are glutathione signals. And in the unlabeled animals, there's no signal. In the MIST labels animals, there's signal for each of those 3 isotopes. And so that was a very important thing to establish, that those channels, those M/Z channels are clean. So we do have some overlap of isotopes from the natural form, and we can easily make a correction matrix based on the abundances of carbon-13 in the environment. We build a percent isotope enrichment tool in MSI Reader, and then we can actually do a correction and look at the MIST labeled liver samples versus the unlabeled. So this is just showing you the GSH that's unlabeled, and this is showing you a MIST labeled tissue sample. And again, we see no evidence of, in the unlabeled animals, that there's any crosstalk between the different M/Z channels. So then we wanted to extend this on. So Allison and then a newer graduate student, Russ Kibbe, took this on, and we wanted to start getting into mouse mammary tumors. And so this is just a picture of the tumor that we used. This is an H&E stain. You can see this region right here, that it's necrotic tissue. Up here, it's a muscle, it's non-tumorous. And then we have some regions where there's tumorous tissue. And so this is the normalized glutathione abundance. And you can see right away in glutathione that's unlabeled is that there's no glutathione signal where the necrotic tissue is. This is the pi labeled stable isotope labeled 1, 2, and 3, and you can see as you go on in time, of course, you're going to produce less of the stable isotope labeled 3 because it hasn't as much time to incorporate into the GSH. But nevertheless, it gives us some idea of the kinetics, whether it's going up, down, or staying the same. And so this is in the muscle region. What you end up seeing is high steady state glutathione with very low enrichment rates. Down here in the tumor, red circle, this is high steady state GSH with high enrichment. And then in some regions that are marked in blue, these little blue dots here, this is decreasing enrichment where it's basically washing out the signal. And so in these 2 studies, one was the liver, one was the mammary tumor in the mouse. We've demonstrated that MIST works really well, and we can visualize the timed stable isotope label enrichment per voxel to reveal the rates of synthesis in the proliferative tissues. And so we're extending this on to much more complicated experimental designs and following on with these type of experiments. So the second story has to do with a collaboration with Dr. Shen at Duke University, and this has to do with glycerate and how it is, because glycerate is metabolized is produced by metabolism of fructose. And so the idea was if you have a high-fat diet and you're also in consuming fructose through like soda, in the intestine, it gets metabolized, you produce glycerate, and the hypothesis was that it damages the pancreas islet cells. And so that was the working hypothesis that we thought might be true. And so we have a high-fat diet mouse. We have 24 mice in these studies. It's a high-fat diet with a stable isotope labeled drink of fructose. And the no gavage at time point 1 is basically our control experiment because it hasn't had time to metabolize in the intestine and so forth. And then we have a gavage at 45 minutes and a gavage at 60 minutes. And then over here, the control diet is not a high-fat diet, of course, and we have the same time points. Then what we do is we harvest the animals, we take the pancreas tissue, and we image it with IR Moldesi. These are the H&E stains showing you the high-fat diet and the normal control diet, and you can see these islet cells. These islet cells are the ones that excrete insulin, and they are about 100 microns in diameter, and they're scattered throughout the pancreas. And so when we look at the stable isotope labeled fructose, so the stuff that makes it all the way through, we can actually see stable isotope labeled fructose in the pancreas tissue. And this is in the high-fat diet. You can see it time 0, of course, it hasn't had a chance to get there yet. At 45 minutes and 60 minutes, you can see a lot of stable isotope labeled fructose in the samples. In the control diet, you also see it. It's less so. And so we're trying to figure out the mechanism of that. But in the production of fructose, like I said, it gets metabolized into glycerate, and so we wanted to find out if in the high-fat diet versus the control diet, if more of the islet cells are being produced with, or if more glycerate is being presented there in those islet cells. And you can see these little blue dots here in the tissues, and in the high-fat diet, there's a lot of islet cells that have stable isotope labeled glycerate. In the control diet, few and far between. There are some, but there's a lot less. And so when we look at the average detection frequency of the stable isotope labeled glycerate per unit area with the high-fat diet at time 0, 45, and 60 minutes, you can see this rises up very quickly. And so there's a lot of stable isotope labeled glycerate there, and in the control diet, much less so. And so the idea here in the conclusions is that the increased fructose derived deposition of glycerate in the mice fed a high-fat diet resulted in pancreas islet cell damage. It induces glucose intolerance, and this establishes the first link between dietary fructose and diabetes that is modulated by dietary fat. And so there's a tremendous amount of data in this cell metabolism paper. That's a lot of biological studies and so forth to support all of these conclusions. This was the part where we did with the stable isotope labels. So I encourage you to look at that paper. So the third part of the story is taking a big step back and saying, well, you're doing mass spec imaging and you think about quality control and system suitability testing. And the question is can we develop a methodology for mass spec imaging? And so they're essential for monitoring existing variability to verify experimental results. In terms of keeping track of the state of the system, the repeatability within a day, or the reproducibility between labs and so forth. And so we wanted to see if we could come up with a way to do this for mass spectrometry imaging. Of course, many of you do LCMS, and these are some really nice papers on quality control in mass spectrometry literature. You do see from the year 2000 to 2023, it is a growing interest because people need to figure out if their instruments performing equivalently as they do these larger and larger scale biological experiments. In the mass spectrometry imaging community, there's no standard practice or protocols. There's a lot of variation in ionization sources. Each of those ionization sources have different characteristics of the data. And in mass spec imaging in the field, we primarily rely on MS1 data as our initial go-to. And there's no commercially available mixtures. So an undergraduate, Olivia Dioli, who's in the group, and a graduate student who's moved on, Helena Bai, and our statistician, Emily Hector at NC State, we came up with this idea of trying to use a QC sample. So we have an instrument suitability versus number of samples that we're looking at, number of tissue samples. And so on this slide, we have a QC sample, then a tissue, then a QC sample, and a tissue, and so forth. And at the beginning, the instrument's working just perfect. We know it's working well. We can use the QC sample to check that. So we fire the laser. It's a 20 by 20 ROI. Then we move on to the tissue and we say, yep, it's working great. The QC checks out. But then we eventually get to the point where this is a split axis here. And maybe by the time we get to sample 30, you can just see the signal has dropped significantly. And as you get to 35 and 40, and this is real data. And so then you do the QC check after sample number 29, and it fails. And so this was using a single molecule called fluconazole, an antifungal agent. And so we just wanted to see if we could get this general concept to work. So then you stop the experiment, you diagnose what's happened, and you clean your instrument. But we wanted to make this more of a community-based approach. And so we went on to look at the MALDI-2 literature, the DESI literature, the MALDI literature, and so forth. And we found 5 compounds. Fluconazole is still one of them, but we added 4 additional compounds, so we're not relying on just one compound in our QC mix. And so this is driven now by Alex Sohn and Russ Kibbe in the group. And so we wanted to see if we could make this a really good QC standard. So what we do is we spray a slide with a TM sprayer and store the glass slides at minus 20 degrees until analysis. Then we do the MS analysis, and in this developmental study, proof of concept, we did 118 ROIs when the instrument was clean. When we knew the instrument was compromised, we collected 68 ROIs. And then after we clean the instrument, so hopefully it's back to the beginning in terms of its initial conditions, we collected 10 ROIs. Then we do data extract and MSI reader. We do principal component analysis, which is scaled and centered. And then we do develop an SST score that describes numerically and objectively the state of the system. And then if we need to, we can do troubleshooting measures if we have to intervene. So this shows you the data from the 5 different molecules that we chose in this initial study. And you have 5 different molecules. They're equal molar concentrations, but of course their ionization efficiencies are different. So, but they're relatively present at the same abundance. This is showing you the ROIs from day 1, 2, and 3. So you see caffeine in day 1 and it's shown there in red, and then green, and then purple. And these are the 5 different molecules. And you see some variation in there. And that's actually very important because the absolute abundance in a mass spectrometer is going to change, right, within a day, a week, or whatever. And so we want to capture that natural variation of the abundance with the exact same samples. And so we know what's to be expected when the instrument's performing properly. So we do a preliminary screening of the data. This is just showing caffeine. We look at the detection frequency, the monoisotopic mass. If that fails, then we just stop the experiment because the detection frequency we know should be above 98% and in this 20 by 20 ROI. And if it's not, then the experiment's already over because there's something really wrong with the instrument. If it passes the detection frequency filter, then we use mass measurement accuracy. And right now in our lab, we're doing mass spec imaging with sub ppm mass accuracy. So we set our tolerances at 1 ppm. Then we look at the abundance to see if the caffeine peak is equal to or above this threshold, which is shown here in green. Then we look at the spectral accuracy, looking at the C13 isotope to make sure that we're counting the correct number of carbons within reason. And then we look at the isotopic detection frequency. So we take the A+1 peak, which is much lower in abundance, and that actually becomes very useful to look at the detection frequency of that in building the model. And so then we can use all these metrics to calculate a final score. So the way this works is you have a 95% quartiles, the 2.5 and 97.5 quartiles or quantiles of each principal component per analyte. And this is what we really care about. This is just showing you some of the data from the PCA. We do this, then the second part is we define the variance explained by each principal component and for the first 4. And you can see they kind of follow the same trends, but they're a little different. But that's a good thing, again, because we're capturing more of the reality of the system. And then we can test these ROIs in the model and see if they actually work. What we have to do then is we have to calculate the D of each principal component, which is the number of values within this range divided by the total number of scans. And in our case, it's 400. And so this is, in this case, we have 280 that fit within this quantile range. And so that gives a DPC of 0.7 as our weighting function. And then we have the PCs giving us our loadings for each molecule for caffeine for each of the 4 principal components. We calculate that for the analyte. And then for caffeine, this is just showing you one example. So the SST for caffeine is 0.76. What we can do then is do that for all 5 molecules and divide it by the number of analytes. So the SST final score scales between 0 and 1. If one of these molecules doesn't work in your ionization technique, you just don't use it. And then you can actually just divide by 4. So it'll be more universal than just for our lab, of course. And so we have to reach that SST threshold, and that's very important. And if it's not, then we want to, we have to intervene with the instrument. So where we're going with this right now, we're completing the current model. We're characterizing the acceptable values for detection frequency and establishing a statistically determined cutoff threshold for the final SST score. So that's almost complete now. But moving forward, we think there's a lot of value in using native and labeled analytes, because in our technique in particular, we could monitor the electrospray stability here, for example. So caffeine was one of the molecules here on the glass slide, but we could also put caffeine in the electrospray emitter as a stable isotope labeled version, and that would allow us to monitor the ESI stability over long periods of time. We could also use the ratio of native to labeled species to characterize the ablation/ionization efficiency via combined number. And then we want to incorporate this into the QC SST model. And that might vary a little bit between different mass spec imaging ionization techniques. So this is kind of where we're going right now. We're finishing this, and then we're going to move into stable isotope labels into our lab. So this work was, I always give all the credit to my students, because as John Fenn taught me a long time ago, that I learn more from my students than they'll ever learn from me, is what he told me. And that couldn't be more true. So not only the students, but all of our collaborators, in this particular case, Emily Hector, our statistician collaborator. So with that, I'd be happy to answer any questions.
Thank you, Dave, for that excellent talk. We have time for maybe one question. I don't see any from the floor, so I'll ask one of mine. What was the criteria used for the SST metabolite panel selection? And as a two-parter, the second half, have you preliminarily applied that to track QC processes in pooled or individual sample analyses?
Yes, that's a great question. So the first part of that is we started with fluconazole. It was somewhat random, but we had to find something that wasn't endogenous to the type of samples we look at. So, in mice studies and mouse studies and things like that. Then we extended it to 4 additional compounds, and we wanted to find one with sulfur in it. So we used an HIV drug. We wanted to have a variety of molecular weights, but you notice we went from caffeine. I think the highest molecular weight is fluconazole. So it's in the 300s. It's too narrow of a molecular weight to really capture what we need to do. So we need to rethink the molecular weight range that we're capturing in our system. So it wasn't arbitrary by any means, but we wanted to have some compounds with sulfur. We wanted to have some molecular weight diversity and some chemical structural variability. But we really do need to rethink that. So right now we're at the proof of concept. We did, using just fluconazole, do studies where we would monitor the SST score that we built just in preliminary studies, watching the instrument decay. We would do fluconazole once every week and do a bunch of ROIs, and for sure we could actually show the SST score drops really quickly when the instrument's right at the limit where it's no longer functioning properly. And we validated that by looking at the tissue data that bookends those QC samples. So it really does work, and we've tested that. It's just we wanted to move on to a more diverse panel, and now we want to make that panel even larger in terms of the molecular weight diversity and the availability of stable isotope labels, right? Because we have another criteria that we have to pick molecules that have a stable isotope label equivalent. So that's where we're at.
Well, the 5-plex is a great starting point, and it's sorely needed in the field. So thanks again, Dave, for that excellent talk.
You're welcome. Thank you.
Now move on to the second speaker.